My name is Melanie Philofsky, and I'm moderating the lightning talks this afternoon. Our first speaker is Martin from the Department of Medical Informatics at Friedrich Alexander University in Nuremberg, Germany. Mehr Kashyap brings together 18 institutions throughout Germany. They have adopted the OHDSI and OHDSI tools to harmonize their disparate terminologies and facilitate research. After that will be Maxim. In recent years, he has been involved in several OHDSI projects at the Hive, a company specialized in open source solution for bioinformatics and translational medicine. These projects were aimed at converting electronic health records from different European countries to the OMOP Common Data Model. Specifically, the Hive created a transformation of Swedish registry data, which will be presented here. Nicole Pratt is an associate professor at the Quality Use of Medicines and Pharmacy Research Center at the University of South Australia. Nicole's research interests are in the development and implementation of novel statistical methods to monitor the safety and effectiveness of medicines and medical devices. Mehr is a medical student at Stanford University. He is currently working in Nigam Shah's lab developing prototype classifiers for various diseases. Previously, he studied computer science and cell biology as an undergraduate at the University of Rochester. He is broadly interested in using machine learning tools to power clinical research and improve patient outcomes. Dr. Regina Boland is an assistant professor of informatics at the University of Pennsylvania and has collaborated with OHDSI for the past 3.5 years. She will be presenting her work for us today involving uncovering exposures at birth and their effects. Rohit comes to us from Stanford University. He uses observational data in the OMOP CDM from over 100 million patients in 4 different countries to examine effective clinical treatment pathways for type 2 diabetes. Our last speaker comes from South Korea. Chan compares Korean national health insurance data from the OMOP CDM against American and European guidelines for initial combination treatment in high-risk hypertension patients. First up is Martin.
Thank you. Good afternoon. I'm Martin, and I'm one of the new guys from abroad saying hi to the community. Hi. We all know that we desperately need data to do research, and access to the data is not an easy endeavor. Not only hampered by different data formats and different vocabularies, but also by different access policies in different institutions. And America is more advanced, I guess, but even the German government recognized the problems in the situation, and they are going to spend 150 million euros, which equivalents $180 million, during the next 4 years in order to enable, especially university hospitals, to build up so-called data integration centers. So to collect all the data that a patient generates during the stay in a hospital and to open this up for care and also for research. And they also want to harmonize use and access policies and all these topics about data privacy and so on, to build a national highway for medical data to foster research. They all know that this also means we need people knowing what they do, so they also invest in education. So for the last few months, consortia were asked to develop concepts on how to do this, and now 4 consortia will be funded with 120 million during the next 4 years. The rest of the 30 millions are spent on young researcher groups. It's expected to be about 30 or 40 new professorships to be funded in Germany. So we hopefully will hear a lot of new research in Germany in medical informatics during the next years. So Miracum is one of the 4, and it's the largest, by way the largest consortium, which is coordinated by our department. We already have 8 university hospitals, which is a quarter of all university hospitals in Germany, with 10 million patients and their data. and more hospitals to be included during the time not all the hospitals received funding. When building a data highway, we want also to show that the highway is beneficial for care and research. And so all the consortia are asked to look into 2 or 3 use cases, which in our case range from the more traditional feasibility, secondary use topics that we, for example, have experience with I2B2 and Shrine, but also into predictive models, machine learning models, molecular tumor boards, in which with usability issues, the added value of harmonized data can really be shown for the benefit of patients. And in contrast to other consortia, we do not believe that a commercial vendor is able to deliver on the tool, a proprietary tool, which solves all these problems. We built on the strong communities that have proven during the last years that they built wonderful tools, and one of them is OMOP and the OHDSI tools. And so OHDSI and OMOP will be one of the pillars in our research in networking the university hospitals in Germany. And we were not only talking about concepts during the last 9 months, we already established a proof of concept study in Germany with our partners, and we were able to get them with a virtual appliance with all the tools installed to import their claims data into the database within 3 months' work from the idea that to do such a study into when we were able to run a federated query. And for example, we visualized the catchment areas of our partners, or we did a study on the treatment schemes of colorectal cancer. Just a proof of concept, no medical innovation here. But it shows it is possible if all partners are willing to do it. So this was my chance to say hi to the community. George Ripsack is one of our scientific international advisors in Miracom. So this is also why we looked into OMOP and OHDSI community, and we are very happy and would like to be part of that. We would like to import all our clinical data. We are more advanced in-house than in the network, but you will see it in the next few years. We would like to collaborate on the vocabularies. We use some different vocabularies in Germany that are not yet available in the OMO vocabulary or in Athena. And, but if you invest in that, we share this, of course. We are not yet allowed to use NOMID in Germany, but there is a national license. So there are many things going on, and we will keep you updated on that. And I already took the chance to have some talks. We are building an ecosystem with OMOP on Fire and Smart on Fire, and have variables with a Fire interface via OMOP on Fire into this database. So we are really interested in getting all the bits and pieces done to deliver an ecosystem and not only a collection of tools in here. And if you have any questions, do not hesitate to get in contact. Thank you very much.
Hello. In the next few minutes, I will be telling something about the transformation of Swedish EHR data to the common data model, which we developed at the Hive. I'll first focus on the vocabulary mappings that we made, and then go into detail about some important ETL decisions, if I have time. So the vocabulary mapping was of vital importance for this project. Besides some small code lists, there were 4 main source vocabularies, for which each the Swedish concepts had to be mapped to the standard OMOP concepts. There was a Swedish dialect of ICD-10, the NOMESCO classification of surgical procedures, the Swedish National Drug Registry, and some local specialty codes. None of these were already included in the OMOP vocabularies. If you want to know more about the technical details of how we did this mapping, some we could do automatic scripts, and some we had to do manually, one by one. If you want to know more, I would like to address that during a break. But now I would like to focus on how we assessed the quality of these mappings. So this was really important, especially because we did the ETL in several iterations. You want to know what the quality of the mapping is at each phase. So for this, we calculated the coverage of each vocabulary mapping. And here you can see, for instance, that for the NOMESCO mapping, NOMESCO vocabulary, we could map 13% of all procedure occurrences to the Omicron data model. And it was because we just mapped a small subset of the 6,000 SNOMED codes. The local specialty codes, which also had to do manually, we could map almost all of them because there was just a small set of codes. There were just 200 codes. However, this does not tell you everything about the quality of these mappings. For instance, for the drug mapping, you can see that just 1% of the drug exposures were not mapped. So that information is lost. But this is just the missing mappings. If we zoom into this drug mapping, we can see some drugs are mapped to the clinical drug level, some to the ingredient level, and at each of the levels, some information is lost. For instance, this large portion, the clinical drug, there the brand information is lost. And some of the source concepts actually had this brand information, so that information is lost. And for the other pies, more information is lost. For instance, for ingredient, the dose form, and the strength information. Still, this does not say whether the mapping is correct or not. And therefore, we considered some ways to assess the quality of the mappings. One of these is a standardized expert review. Already off? Or? Okay. It would be really great if such expert could indicate whether mapping was lossless or mapped down, mapped up or down in hierarchy. Another metric could be the percentage coverage to gain. For instance, this NOMESCO mapping that is shown, which had just a 13% coverage. Well, this could be increased by 14% if the top 10 unmapped concepts are mapped. So there's a lot to gain there. This could be interesting for the next iteration of this mapping. And to wrap this all up, we've made a mock for a quality scorecard, just like a traffic light, where green indicates this is good, this is high quality, good coverage, and red indicates that there's something to be done. It's important to report on, and there's actually, in the near future, some funding opportunities from the EMIF project to further develop this. Then some ETL decisions, 4 short points in the last minutes. As usual, there are a number of mismatches between the source data model and the target OMOP model. And these, I would love to address these as well in the Themis work group. We'll go through them one by one. First of all, the source data includes multiple causes of death. The death table in the OMOP data model has just one primary cause of death. So we start the others as observations. In addition, the source data included circumstances like alcohol-related deaths or work-related deaths, and also the place of death. For these, we did not find a correct place for in the OMOP common data model, so we stored them in a separate addendum table. Next, we found that there are 2 valid ways to store certain observations. An example here, I give the marital status. First, we map them as an observation and as a value in the observation table. Then we store as observation the marital status, and the value is the actual marital status, for instance, that the person is married or not. After running these, we found out that might not be the best way, so we store them just as married as the observation, and then the OHDSI guidelines instruct you to place yes to a positive concept in the value as concept ID. Lastly, we missed a few visit attributes. One of these was whether a visit was planned or not. So we stored it separately as an observation. And also 2 other visit attributes. It was the admitted from and discharged to concepts, so the place where someone was coming from or going to. And actually, these are now included since the last release of the Common Data Model. So we're really happy with that. Although we do have to adjust the ETL again now, this has come up. Thanks for
Okay, good afternoon, everybody. What I'm going to do today is present to you a study we're hoping to do. It's in the planning stages at the moment, and show you how I think, at least, OHDSI is going to blow this thing out of the water. So, this is a feasibility study of the utilisation and the safety of biologic medicines. Biologic medicines are immune-based therapies. They're used to treat cancer, diabetes, multiple sclerosis, lots of different conditions, and in particular autoimmune disorders such as rheumatoid arthritis. So they're clinically effective, they work really well, but they have a unique mechanism of action, and they can result in some unpredictable and life-threatening, some quite serious adverse events. There's expanding indications of these medicines, there's lots of off-label use, anecdotally. People use lots of them. There's multiple use of biologics, and they're used in different treatment pathways. The pre-market clinical trials of biologics, as we know, are very limited in their size, and some of the really serious safety concerns have really only come to light once large populations have used these medicines in the post-market setting. In particular, a review of 174 biologics actually found that almost 1/4 of the products had some sort of safety-related concern that was raised after marketing. So there's a couple of stories that set up the scene and why we want to do this study. So in South Australia, where I'm from, we had 56 patients who were dispensed ipilimumab in our public hospital, and they were dispensed for malignant melanoma. 8 of those patients were actually admitted to hospital for severe colitis. 2 of those patients received a total colectomy, after another biologic, infliximab, failed to resolve the colitis. So this is a very small study, but a very severe adverse event, and cost a lot to the public health system. In practice, the rate of colitis that we saw just in South Australia was 3 times higher than in the clinical trials, and the estimated cost was over $400,000. Another story, eculizumab, hard to say, for treatment of psoriasis was withdrawn 6 years post-market after cases of PML were detected. I'm not going to say that really long word, but it's swelling in the brain. Another drug, natalizumab, also has been associated with that same risk. And interestingly, the risk was increased with increased duration of the biologic, and was modified with prior use of other immunosuppressants. So you can see that the use of biologics is very complex. They're used over a long time, used in different ways in different people. So we need to examine the safety of these medicines. We need to identify the risk, we need to quantify the risk, and we need to determine who might be vulnerable to that harm. So we set up a study. I've written a grant application to our Australian government in February 2017 with chief investigators from Australia, but also associate investigators from the Asian Pharmacoepidemiology Network, which I've been a part of for quite some time, and OHDSI collaborators. In July 2017, we got reviewers' comments, and these reviewers' comments really highlighted how this evidence is real world, and it's going to support clinical decision making. They identified it was significant and innovative, and they thought it would produce globally relevant results. They also highlighted that it was really important to do this study. There was a call to action to look at the safety of these medicines by the EMA, and use of these biologics is increasing. It's really important we understand. So in July 2017, I also got bad comments. I got two key weaknesses, and the first is that the actual number of people on each biologic was unknown in the application, and they didn't understand which side effects we could monitor using these data. So on July 6, we set OHDSI into action. We posted a SQL query on the OHDSI forum, which we heard earlier today, and thanks to Martijn for creating this little SQL query with the help of Patrick. And overnight, while I slept, this is what happened. We had 15 different databases answered the call, and over 7 million biologic drug errors were identified in those 15 different databases, all while I was sleeping. And if we put that into context, the median number of subjects included in pre-market trials is between 438 and 1,700, so really, really small. So I let the reviewers know just how many people we would be looking at. So what's next? We're going to utilise all of the tools of OHDSI to do this study. We're going to look at population utilisation. Who uses biologics? How much do they use? What are the treatment pathways of biologics? Who uses what and in what order? And can we identify different treatment patterns in different countries? And that may help us with our safety studies. We're going to look at population estimation. What is the risk? And we're going to look at patient level prediction, trying to identify those at increased risk. And the protocol will be coming soon on the wiki that we saw. Thank you.
Hi everyone, my name is Mehr Kashyap, and I'll be talking about high throughput phenotyping using imperfectly labeled data. So to get started, phenotyping is the task of identifying patients with a given condition of interest. When you apply that to electronic health record, that's called electronic phenotyping. So there are two primary ways that people are going about to do this right now. So there's rule-based phenotype definitions. These are if, then, and, or queries, basically. They're inclusion and exclusion criteria that clinicians or experts in the field come up with to define a specific condition. So type 2 diabetes definition might be something like, takes metformin, has a SNOMED code, ICD-9 code of 250. The problem with these are that it actually takes quite a while for a phenotype definition to be curated. It requires a lot of consensus generation. The other approach to phenotyping right now is using supervised learning techniques, which is great because your learning algorithm can figure out what the important features are and then figure out who the type 2 diabetics are, or whatever other phenotype you're looking at. The problem with this is training the actual algorithms requires labeled training data, and that's hard to come across, or it takes a while to actually go about and label data. So previously, we've shown that you can take imperfectly labeled training data and train actually pretty good phenotype classifiers. So the labeling function can be something like, look for ICD-9 codes of heart failure and other related codes, and then use those as your labeling function, and then train your model on top of that. That expedites the process of having training data, and so you can use your supervised learning techniques and then train and make a classifier. So Juan Banda, who's actually here today, developed our package that does exactly that, the imperfect labeling and then the model generation. And it's built for use with OMOP common data model. So if you have your data mapped to OMOP, you can go ahead and start building classifiers. And we've actually shown that for type 2 diabetes and MI, the classifiers that were created were just as good as rule-based definitions, which is awesome. So our goal for this particular project was to see basically how many phenotype classifiers we could develop really quickly and have them perform well. So the hypothesis is specifically, can we take a high precision labeling function and then build a model on top of that which maintains the high precision but gives you a recall boost. So you squeeze out a couple more cases with your model. So briefly, the classifier development and evaluation process is this. So you start with an imperfect labeling function. In our case, we look for 5 mentions of the disease-specific SNOMED code within a patient record. And we do this because we really want high precision labeling. We want to make sure the people that we call cases are actually cases. Then from your entire EHR data, you pick non-cases randomly, and then you go ahead and train on that data. So we built L1 penalized logistic regression classifiers on top of that data. Then to evaluate, you use, in the absence of a gold standard manually labeled test data set, what you can do is use rule-based definitions that have already been developed. So there were a couple developed at the last OHDSI hackathon. There are a couple implementations of PKB rules on Atlas available. So you can use these to define your test set, and then you can go ahead and test your classifier on them. And that's what we did. So we ran our classifiers on a test set composed of 10,000 patients. And importantly, the prevalence of cases in the test set equaled the actual disease prevalence. So it's mimicking what a classifier would see in the real world. So we did that for 10 different phenotypes. And what we saw is for 8 out of 10 of the phenotypes, we maintained the high precision that we had in the original labeling function and get a boost in recall. That's actually pretty significant. And this boost in recall can take you from single digits recall to double digits. So for example, if you look at cataracts, the recall with just looking for 5 mentions of the code for cataracts and related codes is 0.04, which is really low and might not give you enough cases to actually go do a large observational study on top of that. But when you build a classifier on top of that, you get a recall boost of 0.22, and you still maintain pretty high precision, so 0.87 in that case. There are other phenotypes in here that perform better in precision, have higher recall boosts. We did see for 2 phenotypes that there was a significant precision drop-off, and we're working on trying to figure out which phenotypes are good for classifiers like these and which aren't. But the nice thing is, for the majority of them, we saw that we can develop these high-precision classifiers and get a boost in recall. And what I think is the awesome part of this is you can build these classifiers really quickly. So building 10 phenotype definitions, the rule-based ones, would take anywhere from weeks to several months. We can build each of these phenotype models in essentially an hour and a half or less. And if you're mapped, again, to the OMOP Common Data Model, you can start doing that at your own site. That's awesome because that allows us to then share models, create them at different sites, validate them at different sites, and that's the direction that we're headed in now. So thank you.
Okay, so I'm here to tell you about my work. I have a lot of slides, and mainly for you to peruse after my talk, because I'm going to have to go through some of them quickly, but I'm going to talk about the more important or more interesting things to greater depth here now. So I first want to define 2 important distinctions, which I think is important in the context of OHDSI. So there's a difference between latent effects and overt effects. And a lot of what OHDSI has done is actually looking at overt effects. So this is whether or not when you have a drug, do you get a rash, do you develop things like edema directly after the drug or within 30 days of drug exposure or possibly a year after drug exposure. So what I'm actually looking at is rather different from what other people have done. I'm looking at latent effects. And by latent, this is defined as variants related to different exposures while you're pregnant and the effects that that would have when your child is 25 years old. So these aren't things like what happens to your child when they're initially born. These are things about what happens much later in the child's life. So I'm specifically looking at birth season-related exposures, and this includes things like pollution. It also includes things like climate factors, anything that would have a birth seasonal component. And we're looking at the offspring risk of disease later in life. So we're very lucky actually to participate in the OHDSI Consortium. We ended up getting 6 data sets through this mechanism. A couple were not actually OHDSI participants at the time, but are subsequently. And you can slice this data in a number of different ways depending on your research interests. So you can look at individual sites, you can look at exposure to Pacific versus Atlantic Ocean, you can look at exposure to specific climate variants. We ended up with 10.5 million patients, and these are the demographics of those patients. And we looked specifically at 2 kind of broad types of exposures. So we looked at cultural effects, when specifically relative age effects. Versus another analysis looking at the climate and pollutant effects. So you may be asking what a relative age is. So relative age is something that is commonly studied in sociology and psychology, and is related to sports and sports outcomes. So people who are born during certain times of the year tend to perform very well in sports. And this is because of very specific, hard deadlines given by international organizations related to certain athletic leagues. And this causes children who are older relative to their peers to do very well. So when you look at a plot of professional athletes in specific types of sports, such as soccer or football, you will see a birth month distribution, which is non-random, and it's actually driven by this relative age effect. So this is very interesting if you're a sociologist, but what's also interesting is how does this relate to birth season or birth month exposure studies? So we had 4 different cutoffs in our data set. We had 6 sites with 4 different school cutoffs. And we actually adjusted them based on this relative age phenomenon. And we looked at whether or not you were 6 months older versus 6 months younger than your peers. And we looked at all diseases, not just one. And we only found one significant result. And that was attention deficit hyperactivity disorder. So this is the unadjusted curve. This is just the raw birth month curves across the 6 sites. But when you look at it from this lens of the relative age effect, you end up finding an extreme correlation between those who are born younger relative to their peers and increased risk of attention deficit hyperactivity disorder. This has been something that's been discussed in single-site studies previously, so we're kind of validating that finding, except in a much larger cohort across many different sites. And the overall effect was, on average, about 18% difference in risk of attention deficit diagnoses based on this phenomenon. After looking at that, we also then looked at what people more traditionally think of as birth season effects, things like pollution and climate. For this, we have a very large method diagram, and then I will go through the different pieces. So first, we computed an average gestation period based on your birth month. And then we computed the cumulative exposure to different pollutants and climate exposures, such as sunlight and precipitation, across the different trimester periods, as well as pregnancy-wide and then perinatal. So we had a bunch of different correlations, and we integrated them using a meta-analysis statistic that's commonly used for combining data from multiple different sites. And this is some more information about that. And I do want to point out something that I get a lot of questions about, is that there was actual variance between the different exposures at the different sites. Some people say, you know, how different was the variance? This is just 2 examples of sunlight and ozone. As you can see, some of them are, you know, bimodal distribution, some of them are kind of more Gaussian in nature. We ended up with pooled site-wide correlations across our study sites. And because we did a lot of comparisons, we also adjusted for multiple comparisons using Bonferroni. So this is a zoom up of just the plot from 3rd trimester exposures. And I'm going to focus on that one little dot. So this is 3rd trimester sunlight and exposure to an increased risk of type 2 diabetes in the offspring. So we found actually that an inverse correlation where low sunlight during 3rd trimester increased the risk of the child's risk of type 2 diabetes. And this is something that actually fits fairly well with the biological mechanisms behind increased risk of type 2 diabetes that's related to the mother. We also found some more interesting things. So this is first trimester exposure to fine air particulates and increased risk of atrial fibrillation in the offspring later in life. And again, this is something that fits with the literature on first trimester exposures affecting cardiovascular disease development and so forth. In summary, I hope I have convinced you that OHDSI can be used for more than just drug exposures. I haven't actually talked about drugs so far. And also the importance of climate and understanding disease susceptibility, as well as the importance of prenatal and perinatal exposures and your risk of disease later in life, and towards a more comprehensive understanding of disease risk, which helps to fit in with some of the precision medicine initiatives. And also the findings results actually fit well with known developmental pathways, and I believe in part this is because of a very stringent Bonferroni corrections that we performed on our p-values to make sure that we were only finding things strongly supported by the data. There are many people that I must acknowledge, including everybody who helped participate, as well as members of my dissertation committee. This work was performed while I was actually at Columbia University Medical Center in New York, but I also must thank my current employers at University of Penn.
Good afternoon, everyone. Today I'll just present our work that we've been trying to do by using the OHDSI network on learning effective treatment pathway for type 2 diabetes. We all know that diabetes is a major problem, both for our patient and economical burden. And we'll see that how we can utilize this OHDSI tools to learn effective treatment pathways for that. So as far as the guidelines are concerned, for the treatment of type 2 diabetes, that once a patient gets diagnosed with T2D, the doctor says that you go on and start prescribing metformin. Once the metformin start demonstrating an adverse effect, then there are multiple options that one should choose from. Then there was a recent study that was undertaken by OHDSI last year, where they have shown that there's a considerable amount of variations that exist in the treatment pathways, the way doctor actually treat type 2 diabetes. So that sort of brings us to a question that when metformin fails and when there are a huge amount of heterogeneity that exists in the second line treatment option, which one would work better? Like, which is the best second line treatment option to sort of choose for? To address this question, we'll see that how what we have done. So here we have used the data for about 103 million patients over 4 countries. All the data was organized into OMAP, and our study design is basically that we look at the study at one particular site. We use all the tool stack of OHDSI, implement the study, assess the results, and then ship the code to other sites so that people can perform the analysis. We gather the results and then perform a meta-analysis over it to sort of have a consensus effect size. So overall, the study design is that we pick up the patient with type 2 diabetes. We construct a treatment cohort and a comparator cohort. We subject that to, we construct a patient feature matrix out of it and subject to a matching based on the features. We take this treatment and comparator cohort and perform time to event kind of analysis, and then perform a meta-analysis to assess what we're getting. So as far as cohort construction is concerned, we start with a diabetic patient. We consider, for example, let's say when the biogonide was prescribed, that would be considered as index date. And then we follow a patient for about a gap of 90 days, and then the second prescription was prescribed. So that's how we go on and start building the treatment pathways for T2D. Then I'm just showing a result for one of the example of sulfonylurea versus DPP4 inhibitors. And then we perform this time to event analysis, and what you see is that reduction on the y-axis, the reduction in HbA1c below 7% after the treatment. The blue one is your DPP4 inhibitors, and the red one is sulfonylureas, and kind of suggests that DPP4 inhibitors are better off in reducing HbA1c below 7%. We do this same analysis for other different outcomes related to myocardial infarction, kidney disorders, and eye-related disorders. We gather these results across multiple sites which have participated in this study so far, and we perform a meta-analysis over it in order to sort of assess its reproducibility and how much effect that we can infer from it. So upon meta-analysis, we look at there is a considerable consensus effect size with a very well-defined confidence intervals around it. For myocardial infections, we don't see much going on. Like we can't sort of say which of these two drugs would work better in reducing the incident related to myocardial infarction. Similarly, for kidney-related and eye-related disorders, there's not much which is going on. So in conclusion, because I just presented an analysis only for 2 drugs, so what we find that DPP-4 inhibitors are more effective in reducing HbA1c of patients, but there's hardly any effect of them on myocardial infarction or eye-related disorders. And this study kind of exemplified that you can use the OHDSI platform to do this network kind of analysis where you have your defined question. And we have the code open, and if you have your data in OMAP schema, you are more than welcome to sort of join the study. And with that, I would just like to thank the team, the awesome team of OHDSI. So this is basically our lab, Nigam and other people in the lab, team J&J, Patrick, James, Jamie, then IMS Quintiles, Mount Sinai, Cerner Health, Ohio University, and Columbia. All these people have participated. So that's it. Thank you.
Okay, I'm Chan from University of South Korea, and I'm here to talk about the ongoing study about comparison of combination treatment in hypertension. Currently, American guidelines and European guidelines both recommend to initiate a combination treatment in hypertension patients, especially at people are at risk. So 3 kinds of combination regimen are recommended. First one is ACE inhibitor or ARB plus calcium channel blocker. I will call it as AC combination, and ACE inhibitor or ARB plus thiazide diuretics. I will call it AD combination, and calcium channel blocker plus thiazide diuretics, which will be CD combination. But so far, there are only limited evidences for optimal combination regimens in treating hypertension are available. There are 2 kinds of RCTs so far, but they included different kind of people from different areas of world, and the results were different. So, and moreover, there is no real-world evidence comparing treatments. So, we don't know which would be better or the best among these 3 kinds of regimens. So, I want to conduct head-to-head comparison of mortality risk of combination hypertensive medications among patients without high risk for cardiovascular events. So my target regimens were, those are 3, AC combination, AD combination, and CD combinations. I used a National Health Insurance Service National Sample Cohort database, which contains consecutive observation for 1 million patients who were random samples from whole Korean population between 2002 and 2013. It was converted in CDM version 5. I included those who start to use dual antihypertensive medications within 30 days, and with the minimum period of drug for 180 days. And I excluded patients who had ischemic heart disease, heart failure, stroke, or death before drug initiation. The primary outcome was all-cause mortality, and the secondary outcomes included cardiovascular death, newly developed myocardial infarction, heart failure, stroke, or all the composite endpoints. I used large-scale propensity score matching with caliper of 0.15. Max ratio was 1:1, and univariate Cox regression with stratification. For the sensitivity analysis, I conducted the same analysis on the patients with various minimum periods of continuation of the drug, for example, 3 days, 1 year, or 2 years. And for the reproducible research, the analytic code is available on the GitHub in the study protocol sandbox of OHDSI, and you can see the whole protocols in the wiki, and you can see the forums. So there are 3 comparisons: AC versus AD combination, CD versus AC combination, CD versus AD combinations. As you can see in the balanced scatter plot, the difference between those 2 cohorts in the single comparison gets similar after matching. The standardized of mean almost below 0.1. I used 38 negative controls to validate my statistics, and you can see that only 1 or 2 to negative controls turns to positive. So false positive proportion or type 1 error is about 0.03 or 0.05. What is interesting for me is that ACE combinations, ACE inhibitor-based combinations tends to be younger, and after matching, the age distribution gets similar. Most of the patients are in their 50s or 60s. Again, ACE inhibitor-based combinations tends to transcribe later, but it gets similar after matching. Baseline characters is different from the comparisons, but it's very similar between the two cohorts after matching. So finally, you can see that all-cause mortality was not different from among the regimens. So there is no difference in mortality between dual combinations of antihypertensive medications. In the secondary endpoints, I don't see any differences in terms of CCE or cardiovascular death, myocardial infarction, heart failure, or stroke among those regimens. So in summary, there is no difference in reduction of mortality between antihypertensive dual combination regimens in the population with the previous cardiovascular outcomes. And there is no difference in reduction of stroke, myocardial infarction, heart failure, or cardiovascular death among dual combination treatments. This result was presented in the highlight session of European Society of Congress, which means that cardiologists are very interested in this issue and want to find what's the answer about these questions. So we are now recruiting international data partners who will run our NLI code on their database and share the results. It's very easy for you because you have to do is just click the execute function in R, because all the codes are wrapped into the package. So please join my study. Thanks.
All right, great. Now we have time for some questions from the community for any of our presenters. Does anybody have any questions? Please come up to one of the two microphones.
I'm sorry, I don't remember the name of the speaker, but it was regarding the analysis of data predictively in electronic health records and trying to identify if an element was present using a SNOMED code. And my question is, each electronic medical record stores data very differently, and I would want to know how you even replicate this in a different electronic medical record. What was the medical record used in the study? Thanks.
Is that for my project? Okay. Yeah, so the electronic health record that we were using is Stanford's STRIDE data. It's an extract of that data, but all of that data has been mapped into the OMAL common data model. And so the classifiers were built on top of that. So, if your data is also mapped to OMAL common data model, then, we should be able to share models. You should be able to build at your site, that kind of thing.
So Andrew Williams from Maine Medical Center. I had a question for Mary Boland about how the process she used for relating environmental data to the CDM, and selfishly, whether she's interested in contributing to the OHDSI GIS work group's effort to standardize how that kind of analysis is done.
Thank you. So is your question whether or not, so I didn't know there was a work group now. So when I started this project, it was about 2 years ago. So it was a process of recruiting patients, and one of the big hurdles for getting people to participate in my specific study was the requirement of birth month. So a lot of the data sets within OHDSI do not have date of birth or some type of birth month. So that's why it was kind of a longer process than some other people's. And is your question on standardization of environmental data, or is the question how did I correlate it with the clinical outcomes? Sorry,
I guess to the extent that there was any standardization involved for the environmental data and any kind of standardization of the process for thinking about how it relates to the CDM.
For environmental data, I use the EPA data from the United States, for the United States data. For Korea, there's a similar type of system where they monitor the air pollution, and I can't remember the exact name of that system, but it's in the paper. The paper is actually published in JAMIA at the end of September. And for the pollutant data, I used NOAA, which is the National Ocean and Atmospheric Association data, and they have global data as well. Does that help a little bit?
It does. Thanks.
I'm Rosalie Bright from the Food and Drug Administration, and my question has very little to do with my work, but my curiosity. When you determine relative age in the class, how did you know how that was determined? Because how was that determined? Because in my experience growing up, I moved around, lived in a lot of school districts. I was born in October, and sometimes I was among the oldest in the class, and sometimes I was among the youngest in the class. So how did you deal with children's experience changing over their lifetime, and how did you figure out in the first place what their relative age was in their class?
That's actually a great question. So the way it was done was I used actually the public school cutoff dates. So parochial schools actually have different school cutoff dates than public schools typically. So I used the location as well as the public school cutoff date for that location, given that time period. And with the assumption, of course, that people are going to a school, say in the city, which they got medical diagnosis, which is a little bit of an assumption in places like New York City, where New Jersey has a different cutoff point. But on average, we found. So actually, I should point out that in Taiwan, where it's a national standard, the entire country, and they don't have a separation between parochial and public school, the variance was 40% between peak and trough periods, whereas on average across the United States, in the data that we looked at, it was 18%, which is much lower, which could actually be the introduction of error in that calculation. So perhaps if we had your true position in school, we could actually get a little bit better clarity at that.
So you didn't even know exactly which city they grew up in, right? What did you do when people were born in one place and when they're 25, they're living somewhere else?
So for attention deficit disorder diagnoses, we're actually lucky because typically they're diagnosed in childhood. They have a diagnosis, so it's a little bit less of a stretch. So say you're 12, you have a diagnosis of ADHD, and you're in New York City, there's a little bit less of a stretch than, say being 50 and having attention deficit disorder. But there is definitely noise introduced in that calculation.
Yeah, thank you.
Hi, Mark Bino from Case Western Reserve University and the Institute for Computational Biology. My question is for Nicole Pratt, and I'll start with a comment saying that the number of biologics that are currently in development and their ability to bankrupt the entire health system should be of concern to all of us, but do you see your work as perhaps influencing policy on how quickly these drugs come to market, as well as maybe slowing down how easily they're prescribed to people?
Good question. So I sit on Australia's Drug Utilisation Subcommittee, which looks at the utilisation of medicines for subsidy under the Australian subsidy system. And in recent times, I've noticed that most of those new products on the market are biologic. So you're right, there is this increasing use of biologics. I'm not, this project will hopefully provide some evidence for everybody, policymakers, for doctors, for patients, to kind of weigh up the benefits and risks of those medicines. So it is my hope that it will definitely be translated into all areas of healthcare, not just policy, but it's a very, very important point at the moment. Was there another question you had? No?
That was it. Thanks.
Hi, my name is Kees van Bokhoven from The Hive. So I think my question is to Martin, although if anyone else wants to take it, it's fine. You mentioned that your feasibility query experiment was inspired by ITB2. A couple of weeks back, I was at the ITB2 Transmart conference, and there we were discussing how OMOP compares to ITB2, and concluding that they're mostly complementary. But from an OMOP Odyssey perspective, I'm extremely interested to see how we can extend OMOP and maybe use some of the other efforts like I2B2, but also GA4GH, where it was yesterday in Orlando, or even CBio Portal, specifically for oncology, to look at things that are currently difficult to find in OMOP. Take in oncology TNM staging, or the whole genomics area. So, you know, from your perspective, how do you look on that when you want to build out the medical informatics framework in Germany?
This is why I was talking about an ecosystem. So there are so many tools, and CBio Portal, for example, is on our agenda. Transmart is on our agenda. OMOP is on our agenda. And this will be a learning process on how to fit these things together. For example, we are using FHIR as one of, going to use FHIR as one of the backbones for data. We use other tools. We have replicated the very same study I've presented in OMOP, also in I2B2, also to compare this manual community process. I have a research question. Could you please download it and execute it with a version in I2B2, not with a shrine concept, with another concept. Also to be able to compare. It's very early now, but I think we can learn from each other, and it will be a challenge, and we will learn about that. But we are going to, and also the tools provide different tools to different people asking different questions. So it's not one or the other, it's sometimes one data pot delivered in a tool in an envelope that fits to the researcher.
Great, thanks a lot. And maybe we can follow a discussion, or even from this conference produce a paper about how we position these different use cases and data models.
All right, thank you very much, everybody.
Thank you.